Thanks for the invitation to be here. This is a subject that has got a lot of attention from the laboratory community in the last many years. And it's coming. It's hotter now than it has been in a while. Let's see. Right here, I'm currently in St. Louis for about 3 more weeks, and I'll be moving to Phoenix Children's Hospital in October to take over the Department of Pathology there. So I'm also a former president of ADLM, the Association for Diagnostics and Laboratory Medicine. We used to be called the American Association for Clinical Chemistry. They don't allow the presidents to retire with dignity. They keep digging us back out. So that is why I am here. This is probably the most important slide I'm going to talk about today. Dylan talked a little bit about this, about the process of newborn screening. And for newborn screening to work, there has to be a phase during which we can detect and we can treat that disorder before bad things happen. So there's some prenatal testing that's done. There's testing right after birth that gets done. Dr. Guthrie's favorite tool is still the tool we use today, the blood spot card. It is an amazing tool that allows us to measure a number of sequencing techniques, metabolites, proteins, lipids, all sorts of things in that magic little blood spot. There's a ton of information there. Dylan also mentioned there are about 4 million annual births, about 37, depending on how you're counting, 37 conditions in the RUSP. The dirty little secret of newborn screening is not just in the newborn screening lab, and my lab is a key cog. And my lab and many others like my lab are a key cog in that newborn screening process. What we have to do to detect these diseases is this is sort of an idealized picture of the distribution of a biomarker that is normal in a specific portion of the population, but abnormal in a diseased portion of the population. But it's not always, those distributions are not always separate. Often, almost always, they overlap. And when you do newborn screening, the rule is you can't miss any child at all. So what we have to do is we have to sacrifice. We have false positives in that population. It's enriched for people who might have the disease, but there are kids in that population that get referred for confirmatory testing that don't have the disease. And that's our job, is to figure out who really has the disease and who doesn't. And to do that, we use laboratory-developed tests to do that. So depending on how you count, there could be as many as 20,000, maybe 200,000 false positives, depending on whose statistics you like, to get to that 12,500 confirmed diagnoses. Dylan already covered a lot of the disorders that are in the panel, so I'm not going to spend too much time, but I am going to spend a little time about what is a laboratory-developed test. This is my cartoon that I use a lot to explain to people what a laboratory-developed test is. On the far left, in Clea definition terms, there are a series of tests called waived tests. That means they're so idiot-proof that anybody can do them. Think about diabetics with glucometers in their house. They can poke their own finger, do their own test. Think about an at-home pregnancy test. Those are all waived tests. Then there are moderate complexity tests. So the wave test is you drive up to the McDonald's drive-through window, everything is ready for you to consume, right? So moderate complexity tests are more like Hamburger Helper. Most of the tools are readily available. You have to do a little bit of prep work to make it work, but then you can do it. But this also relies on skilled laboratorians. And then there's high complexity testing, which is cooking from scratch. Every piece of dinner has to be sourced and prepped and put together into a system that functions and everybody's happy at the end of the meal. That's an LDT, and those are the things that I'm going to talk about today. So there's this regulatory conundrum in our universe of Clea versus the FDA. What the FDA is really good at is this stuff on the right. So this is a chemistry platform that we have in my laboratory, and there are hundreds and thousands of copies of this that exist on the planet. They are marketed and sold worldwide. The FDA, the manufacturer of that piece of equipment, works through the FDA to make sure that they can make that instrument the same way all the time and have proper process controls, quality controls in place so that that thing works wherever in the world it goes, okay? Clea, on the other hand, is more of a local regulator. Clea develops, this is my test here. This is not over here. This is my test. It's a single instrument. usually. Sometimes it's 2, depending on how important the test is. We have to have it present in duplicate. But Clea regulates my local ability to build and do that test, and it's not widespread. The test that I do in the St. Louis region at this point. I don't do it for every kid who gets flagged on a newborn screening program. Every state has multiple laboratories that do this, and Clea is responsible for how we build those tests, how we monitor those tests, and how we correct mistakes in those tests. So there's a thought that LDTs are not regulated today. That is not true. We have lots and lots of regulation in our world today. These are some of the things that we have to do to build a test. We have to look at the accuracy of the test. How precise is it? How often do we have to calibrate it? How do we calibrate it? What is the smallest amount of a substance that we can detect? And what is the range of the substances that we can detect? These are all studies that we have to do today. These studies take weeks, months, years to accomplish and do properly in some cases. So, and we're caught in between the FDA and Clea here. Clea wants to regulate, Clea does regulate today, and the FDA would like to do more regulation in that space. But I'm going to tell you why that's a problem. So this is the final rule of the final FDA guidance rule comes down, and this is just a really brief description of what we are facing at this point. By next spring, we have to have these requirements in place: medical device reporting, complaint files. We don't do any of this today, and we're given less than 1 year to make all of this regulatory apparatus happen. Then in 2 years, some more requirements come down the road. And in the latter years, what we'll have to do is we'll have to send these LDTs through an entire FDA approval process, which takes a minimum of months and more likely years. So the innovation and the nimble sort of nature of these tests will be defeated in that sort of environment. There are exceptions. The FDA has graciously allowed us some exceptions here. The VA gets an exception. The DOD gets an exception. That doesn't help us in the newborn screening world. Forensic labs get an exception. That doesn't help us. Pre-1976 tests, there are a few of those left, but none of those are the ones that we do today. The HLA laboratories got a pass. I don't know why. The FDA says that those tests are life-saving, and they absolutely are. But many of the tests that we perform in the context of confirmatory newborn screening are also life-saving. MCAD deficiency used to have a mortality rate of 60 to 70%. Today, with proper newborn screening and proper confirmatory testing, that mortality rate is 2.5%. So it's life-saving by anybody's definition. They also will excuse unchanged LDTs that existed prior to May 2024. The minute we make a change, though, we're done. We have to go into the FDA regulatory system. The New York State approved tests also get a pass. I don't think I have a lot of time. That is a CLIA-centric sort of additional sort of qualification process that laboratories can go through. And then unmet needs, which is probably the most important one for us here. These are unmet needs, but the FDA says we can only test for those patients within our own healthcare system. So I test regionally. So that's going to exclude a fair number of kids in my geographic region that don't happen to be a part of my particular healthcare system. So that doesn't help either. So these are the consequences. We are now LDT manufacturers. My name is not on the New York Stock Exchange yet, but there's new regulatory language on top of an already immense amount of regulation that we have. We'll have to learn to talk to the FDA. We don't talk to the FDA very much. We don't know their language. We don't know their processes. We'll have to learn those. New staff in our laboratories, absolutely going to be necessary. Who's going to pay for that? My hospital's not going to pay for that. Fluid FDA rules. The FDA has also said they can change the rules whenever they want to change. So we're going to make thousands, hundreds of thousands of dollars, if not millions of dollars in investment, and then the FDA can pull the rug out at some point. So many labs will stop doing this. I'd like to think the mission of our lab is to do this stuff. So we will fight tooth and nail to continue to do it. With the resources that we have. Other labs that do continue are going to have a lot of extra costs. And here's some of the extra costs. These are FDA user fees for the next fiscal year. Just to play in the game, it costs you $9,000. A 510K, which is the simplest of these sort of FDA submissions, will cost you $24,000. a de novo submission. So a 510K is if there's a device already FDA approved and your device looks like that, you can market it. De novo submission is there's no predicate, but it's a low-risk sort of device. So the studies that you have to do in the submission you have to do are a little enhanced. That costs you $162,000. And then there's the full-blown PMA, which is a big clinical trial that involves thousands of patients, and many of them have to have the disorder that you're trying to test, so you can already see the challenges in rare diseases. Where are those patients going to come from? Many of the disorders we test for, we have 2, 3, 4 kids in our patient mix who have this disorder. How do you do a big clinical trial on that? That costs you more than $500,000. So for me, $500,000, that's like 2 or 3 instruments that I can use to do this testing. So here's the summary and the outlook. LDTs populate a really important cog in going from the screening result to a diagnosis or not, okay? LDTs are already heavily regulated. New FDA regulations are going to duplicate a lot of what we already do. Many labs will stop. We've polled a lot of our membership. It's very clear that many of them will not have the resources to continue to do this. Costs for labs that stay in business will go up. Creativity and innovation in the LDT space is going to suffer, I think, undoubtedly. And we have been ADLM and others have been advocating for a dialogue so that we can, this sort of discussion has happened off and on for the last for 2 decades at least, and at least in my period, my experience doing in this business. So we need to settle on what's the right regulatory sort of environment for these things and not be faced with these sort of cataclysmic sort of proposals that pop up every now and then that threaten the infrastructure for us to be able to do these things. So that's what we're trying to get to. We're trying to get to a happy place where I don't have to worry about this anymore, and I can go about the job of taking care of kids. I'm a dad and I'm a laboratory director, so I care. Thanks.